Okay, well, moving on, let's talk about control of the cell cycle and ways that we can stop the cancer process, or how cancer can take over the cell and cause all kinds of trouble. And we know that there are certain mechanisms for controlling the progress through the cell cycle. There are checkpoints, and we have telomeres, the length of telomeres in particular, and we have chemical signals from within and outside the cell. So let's take a look at some of these checkpoints. This is the cell cycle. You can see mitosis and the PMAT, the prophase, metaphase, anaphase, telophase, and cytokinesis. There is a spindle assembly checkpoint in between metaphase and anaphase, and there are DNA checkpoints in G1, S, and G2 phases, so that if there are any problems, if there's any DNA damage, then the cell would then move towards apoptosis and self-destruct. So let's take a look at these telomeres. So the length of telomeres is very important because telomeres are structures at the ends of the chromosomes that shorten with each cell division. And after 50 divisions, the shortened length of telomeres causes mitosis to stop. So that's a way to control growth. However, some things can take over this process. So we think about the failure to stop at cell cycle checkpoints. For example, if there is a mutation in a gene that usually slows the cell cycle, the rate of cell division is accelerated. If there is a failure to pause for DNA repair, then faulty DNA leads to unregulated cell growth. And if there is a loss of control over telomere length, this can cause great problems. For example, cancer cells have telomerase, an enzyme that elongates telomeres. So then the cells will continue to divide after 50 mitoses.